Hello everybody, welcome back to our channel. Today we talk about biology again. We talk about transport. We have two sections, 1 and 2. One is SL and 2 is AHL. As usual, we'll talk about form and function in organism. We will to human as an example and also plants as an example. Also for this transport topics, okay. First of all, we talk about the transport by the capillaries, okay. There's an adaptation by the capillaries about the blood flow, okay. How the blood flow around the body. First, we talk about the smallest structure, the structure of the capillaries. Okay, capillaries are adapted for the exchange of materials between the blood and the internal and external environment. For example, what we learn for the lung, right? Alveoli. Okay, the capillary is for the transfer of oxygen and carbon dioxide between them. Okay, and for this transfer, because oxygen and carbon dioxide are small, okay, so for that part of the capillary a multiple layer of cell is okay. But now if we go to another aspect, we want to transfer the materials, not just oxygen and carbon dioxide, so the capillary has to be even thinner for the material to pass through the waste, nutrients, glucose, big thing, okay, so it has to be one cell layer, so distinguish it clearly, okay. So capillary has a single layer permeable cell wall for the material exchange to happen, okay. And for if they even for the rapid exchange we found it out, they even have some tiny opening and pores, okay, to let the thing go through it directly. Crazy. And then there is no energy in the capillaries, okay. So low active transport, okay. No ATP. And then molecule exchange is by diffusion. You remember it for the lung. By diffusion too, right? Okay, carbon dioxide diffuses from the capillary to the alveoli, and the oxygen diffuses from the alveoli to the capillaries, the herms, right? Yeah, because they have different concentration, the concentration gradient. The same stuff happens when the capillary want to transfer oxygen and nutrients and hormones and antibodies and water to the tissues, okay? And also when the tissue want to release the carbon dioxide and waste to the blood, okay? There's a gradient. Naturally different, okay? But for the waste part, be careful. There's one thing that you need to remember for the uvea, okay? Uvea is a waste, but this uvea is specifically produced by the liver, okay? So only in the liver we will transfer uvea to the blood, okay? Not the other parts of the tissue, okay? They don't have uvea as the waste, okay? So be careful, okay? And also be careful, I mentioned it, oxygen and carbon dioxide are smaller. Multicellular wall is okay. So the single cellular wall is not for the oxygen and carbon dioxide. Okay? And of course, how to make the capillary tube more efficient in the muscle, okay? Increase the density, right? And the amount of blood taken to muscle, more oxygen, more glucose, more carbon dioxide, more waste can be removed. And that's it, okay? And also increase the surface area, right? By branching out, okay? Just like in the lung branch out, right? And that there are three kinds of, beside the capillary, there are two kinds of blood vessel. One is artery and one is vein. Artery is there for the blood from your heart outside, okay? And for the blood flowing inside the blood, we call it a vein, okay? They have different structure. You have to see it, okay? They have thick layers, artery, okay? Because in artery, remember high pressure. In the vein and low pressure, low blood pressure, so it is thin. And also because in the vein and low pressure, so they can have a big lumen. We call it lumen. For the artery to keep it at high pressure, the lumen has to be small. And of course, remember capillary is just one cell thick, okay? That the structure is like this, okay? Each one is the cell, one nucleus, okay? Then the micrograph for artery, vein, and capillary are completely different, okay? In IBDP requirement, you need to distinguish them. Not by the names, okay? No need to remember the external, media, internal, okay? No, not the main point. The main point is the thickness, okay? The explanation is here for artery is flow blood out from the ventricle, okay? And flow return blood to the atria. Okay, that means that it's a flow blood out from the heart and then return the blood back to the heart. Okay, and for the capillary, it's the exchange of material between the blood and the tissue. About the oxygenated or deoxygenated depends on whether it comes from the lung or going to the lung. Okay, and for the capillary, some yeah before going to the tissue is oxygenated. After going through the tissue is deoxygenated. So both appears. Okay, and the pressure remember actually the heart pump the blood out. So it's high pressure. Okay, so the other two is low pressure. Okay, and high pressure, so we need to wear a thick muscular impermeable wall. Okay, for the wing also a thin muscular but and also impermeable. But for the capillary, it has to be permeable and very thin, one cell thin for the nutrient and other ways to pass through. A lot of oxygen and carbon dioxide, remember. And the lumen is level, then wing to keep it high pressure, wider than artery, and very level because it first increase the surface area. And when the backflow of blood, okay, to artery high pressure, low backflow, so low of valves, okay. For the wing is a low pressure where blood flow may be backflow, okay, so it did a valves, okay. Capillary low pressure also. And we talk about them one by one, the structure of the artery and the structure of the wings, okay. Artery transport the blood away from the heart, okay. There are three layers: tunica, external, medial, and intima. You don't need to remember the name, just remember there are three layers, okay. The layer of muscle and the elastic tissue in the wall of the artery help them to withstand high pressure. So remember high blood pressure. So we need to be a thicker layer, okay. And in cold environment, these blood flows can release heat, right? To decrease the heat loss at the skin level. So at the skin level, the arterioles has a very constriction. Constriction means very level, okay? So the blood flow at the skin along the skin is very lower. So that the heat loss by the blood on the skin is low, okay? To maintain the temperature of the body. We call it vessel constriction. For what? For decrease the heat flow, heat release in where? In cold environment. And the method measuring the pulse rate, okay, in the old time you can measure the feeling by the carotid pulse and the radial pulse. Radial pulse is on the wrist, okay, and the carotid is on the leg, okay. This is quite accurate, okay, but not they are different sometimes. So you have to compare it with the digital result. Okay. So sometimes we use a digital way and use these two ways. But now in modern medical hospital, of course, we use the digital way. Okay. Easier to record the data. And structural wing. A structural wing also have three layers. Okay. Clinical, external, medial, and intima. But they are withstanding low blood pressure, so they can be narrower and the lumen can be bigger. Okay. And also the wing has to be flexible because sometimes the muscle will compress it. Okay. And this is the structure of the heart. And now what you need to learn is about the valve. Okay. Focus on the valve. Okay. Focus on the valve, and we talk about the pressure later. So look at here. Valve is closed to prevent the backflows of blood. Okay. And forward blood through the blood pressure gradient. So atria, ventricle, and then arteries. Okay, atria and ventricle are the two chambers. Okay, and they're going through these two chambers and to the atria and to the whole body, and then go back from through the ring. Okay, and then when they how can they move through it? Okay, of course when the blood flow from the atria to the ventricle, atria has to squeeze the blood inside. Right, squeeze it means high pressure. So the ventricle is low pressure. So this bicuspid valve is opened, go down. Okay. And at the same time, when the ventricle want to squeeze the blood to the atria, okay, it squeeze, okay. Oh, and then become high pressure, but the atria is low pressure. So this valve has to be closed, okay, prevent the backflow of the blood, and the blood will go through this semilunar valve and then go to the arteries, okay. And this is the process. So look at the valve. See, think about, okay, if we push from here to there, open, and then the other one has to be closed, okay. So for the valve, we can say that it's very useful, okay. You talk about whether when you will be a high pressure, low pressure, open or closed. Okay. So valve prevents a backflow of water. Okay. Valve maintain the direction of blood flow in one direction, and valve allow the filling and emptying of the heart chamber. And also how to control the heartbeat. Okay, is by another kind of blood vessel surrounding the heart. We call it a coronary ring and coronary artery. This is the artery and the ring to provide release the nutrient and the waste from the muscle of the heart. Okay, and of course with the, in a fatty diet and high ratio of HDL to LDL cholesterol, we lead to a plaque formation in arteries. So we have a correlation is confirmed, but the causality is hard to confirm. So when the cholesterol and the fat deposit inside the coronary artery, okay, occlusion occurs. Okay, and the occlusion occurs that the lumen becomes small or even blocked completely. So the blood cannot flow to the heart. To the heart does not have heart muscle does not have the oxygen and nutrient. So you may feel chest pain or heart attack. And this is lethal to you. You die if your heart can't pump the blood to the whole body. So remember, saturated and trans-unsaturated fat is bad. Okay, remember, saturated fat is from butter. Trans-unsaturated fat is from margarine. Okay, so what's the good thing? Okay, cis unsaturated. Where does it come from? Which one has a high ratio of it? Olive oil. So IBDP commonly evaluate some data to think about the incidence of coronary heart disease. And the concept is very tricky. Okay, we know in science we can just have a strong correlation, even close to one, but we can never say it's a causality, right? Maybe there's some hidden factor make them have a higher correlation. Okay? Even to the extent that we know there's a strong correlation between saturated fat intake and the coronary heart disease, we can just say it is a high correlation. We can't say it's a causality, okay? Because science is like this way. Causality is confirmed, it's absolute, okay? But fallibility, right? So we just have a strong correlation. But sometimes a strong correlation is a good enough indicator for us to have a better diet of not so many saturated fat or unsaturated trans fat. Okay. Human is finished. Now we go to the plants. Okay. Drastic change. Okay. We talk about how the plants transfer the thing. Okay. Transfer what? Okay. First, just transfer water from the roots to the leaf. Okay. And transfer the ions and minerals from the roots to the leaf also. How can it be? How can it happen? Okay, so one is by osmosis. Okay, so they have some special trick. Okay, so the roots has a high solid concentration. Okay, it can be 100 times the depth of the soil. So it means that it has a no water content, right? So by osmosis, the water will go through it, the wood, the surrounding soil to the roots naturally. Okay, only for energy. Okay. It is a passive transport. Okay, and then how about the active transport? How can the woods absorb a nutrient, iron, and mineral to maintain this 100 times as higher, and then they need that energy? We call it active transport. Okay. And then when they all inside the woods, when they then go up, okay, the water and the nutrient move up at the cell wall or move through the cytoplasm. Okay, and unfortunately we have two names. Okay, move through the cell wall, we call it apoptosis. Okay. Move through the cytoplasm, we call it simplex. Okay. You have to distinguish them. Okay, it's in our simplex. Okay. And the problem of excess irrigation, if we have excess irrigation, okay, and then the salinity of the soil will increase. Okay, increase it to such a high level that the difference between the soil concentration of the soil and the woods decrease. Okay, such that osmosis happen not so smoothly. Okay. And the osmosis of water from the soil to the woods will decrease. Okay, so not in the water. Okay, because of excess irrigation. Okay. And then I talk about what? Okay, the water and the nutrient is in the woods. Okay, now, okay, how can the water go up to the leaf? Okay, to the whole plant. Okay, we call it transpiration. Okay, loss of water by transpiration from the cell wall in the leaves. Remember, at the bottom we give the stoma. Stoma will lose water, right? And the water will drain out from the xylem. Okay, And then when the water drinks up from the xylem, it is empty. Empty means a low pressure, right? So low water pressure and the woods is high water pressure. So the high water pressure will push the water up. So we can say this is a transpiration pool, okay? It's due to the transpiration of water from the leaf, okay? So the stem, the xylem has fewer water there, so low water pressure, and it will pull up the water from the woods, okay? So transpiration is the reason, okay, for the water uptake, okay? Very fun, okay? We lose the water, so we can gain it back. Very fun, okay? And then adhesion and cohesion happens. So remember what's adhesion and cohesion? Cohesion is the stick together of the same molecule. So water molecule stick together by themselves just by the hydrogen bond, okay, cohesion. And water molecule also stick to the xylem wall, okay, by adhesion, okay, so can go up, okay, by both adhesion and cohesion. In this part, you remember adhesion and cohesion. And luckily, we learned it before, okay, so you can recap it, okay. Transpiration stream, okay, is formed by the cohesion between the water molecules. Okay, water moves from the roots to the xylem through the adhesion of the water molecules themselves and also the cohesion between them. Okay, and when the water goes up, the other ions, mineral, and goes up together, just like a co-transport. Okay, you can say, okay, goes up together by this transpiration pool. Okay, how to measure it? We call it a potentiometer. We have a system here like this with the leaf, and it is the water source. By the movement of the air bubbles, we can estimate the transpiration. Okay. Of course, there's a limit. Okay, when the air bubbles go to the right hand side, the last scale, we have to push it back to measure again. So we have to reserve here and open there, and then the water go down, and then we push the air bubble back to the left hand side. Then we can continue the experiment with our change of the apparatus, the other parts. Okay. And also about the adaptation of xylem vessel. Okay, xylem vessel is hollow, right? It lets the water to go through it by cohesion adhesion. Okay, so it's a hollow. Okay, well, it's hollow. Then it has a low pressure, right? So it has to have a thick cell wall to withstand the pressure difference. Okay, of the surrounding. Okay, and it also thick cell wall is not enough. We find out is some neckline. Okay, neckline. Okay, when we have a electron microscope observation, we don't need to remember different kind of neckline. Okay, but in the micrograph, you can see different graph clearly, okay? Some are spiral, some are annular, okay? So the xylem. Xylem is hollow. It has to have a fixed cell wall to withstand the pressure difference, okay? It also to have some lignin, okay, to help it, okay? Adaptation of xylem vessel, remember, adaptation of xylem vessel for the transport of water. So transpiration pool, difference of pressure also is to withstand the pressure. It is also hollow, okay? It's withstand the outside too. And what is the micrograph? Okay, for the xylem and phylum in the stem and in the roots, completely different. Okay, so we need to learn to distinguish them. Cross section is always a circular thing, but completely different. This is the cross section for the stem. Okay, later we will learn the cross section as the roots. Okay, in the cross section, okay, in the stem, in the xylem and phylum will become like the xylem is inside and phylum is outside. It's a small part of the stem. Okay, the other part is the epidermis. Okay, the outside layer, and the cambium. Cambium is the newly formed cell. Okay, and then the cortex is open. Okay, and the pitch is inside to support the whole stem. Okay, what you need to remember is xylem is inside, phylum is outside. Okay, okay, 6 xylem and 6 phylum, and then you have to remember xylem is hollow, so big, big. And phylum is a sieve tube, okay, sieve tube, so small tube, okay. This is the way to distinguish between xylem and phylum, okay. And this is I draw it very well here, but actually in the exam, when you see the micrograph, the electron micrograph, maybe it's a little bit confusing. So make sure big one is xylem, small one is phylum. And for the woods, okay, completely different. For the woods, the xylem and phylum are the whole thing of the woods, no other thing. Okay, so xylem, phylum, and the xylem is inside like a cross, okay, and the phylum is outside. It's still inside and outside, but different representation. Okay, and epidermis is outside with the endodermis. Okay, and how to distinguish? Remember, the xylem is hollow one. Okay, bigger, and the phylum is a sieve tube level. So, and this is the best drawing of me. Okay. In the electron micrograph, it is even very similar, not so clear. Okay, see the big one, big one, big one, big one, big one. So the big one is a sinum, small one, small one, small one, small one, small circle. It is a phylum. Oh, that is all. We have the sharing. Okay, remember, it's always human and then plants. Okay, both part you have to understand. Okay, today we talk about transport one. This time we talk about transport two for the HLF. Okay, if you like our sharing, okay, it helps you a little bit. I'm happy. And please subscribe and share with your friend. We are a small channel. We always need your support. Okay. See you for next week. Okay. Biology again. Bye.